Dear student, today we will talk about DNA and chromosome. DNA, that is deoxyribonucleic acid, is present in all the cells of human in the nucleus and act as a genetic material of an organism. This DNA in each cell is packaged into the structure called chromosome. In human, the haploid set of chromosome carries 23 number of chromosome, and which is made up of about 3.3 into 10 raised to 6 base pairs of DNA. So if it is present in the diploid condition, then the amount of DNA doubles, then it becomes 6.6 into 10 raised to 6 base pair. Now beside nucleus, the DNA is also present in the mitochondria, which is called as mitochondrial DNA. And you know that we have seen in the reproduction, the mitochondrial DNA contribute to the zygote as it is present in the female oocyte. If you look at the composition, chemical composition, chemically DNA is defined as the polymer of nucleotide linked together by phosphodiester bond. So here it carries number of nucleotides and phosphodiester bond is holding these nucleotide. Let's see what is nucleotide. So nucleotide is actually made up of three different things. It has a sugar called deoxyribose sugar, which is a pentose sugar, nitrogenous base and phosphoric acid. Now nitrogenous base consists of two different types of bases, purines and pyrimidines. Purines, again, there are two types of purine which are present, adenine and guanine, and pyrimidine there are two types again, thymine and cytosine. Now just look at the structure. Purine ring is there. So now these groups which are shown in the green, okay, they are the one which interact with each other and they form the hydrogen bond. So the hydrogen bond that are present in the DNA are actually pairing between the purines and pyrimidines. Okay. Now when the sugar in the nucleotide, these three things are linked: sugar, base and phosphate. So first the sugar forms the bond with the base. You can look at here. First position of sugar forms the bond with you can say nitrogenous base ring, okay, and this bonding is called N-glycosidic linkage. Now nucleoside once formed, it attaches with a phosphate and form the nucleotide. So the phosphate is attached to fifth position of the sugar, okay. So nucleotide is so called as the base is adenine, then nucleotide is so called as deoxyadenosine or deoxyadenosine monophosphate, because they have one phosphate that is linked to the sugar and base. So similarly deoxyguanosine, deoxycytidine, deoxythymidine. Now this if you look at this nucleotide and if you look at the polymerization, okay, so when the polymerization occurs, look at here, then you can look at 3' position of the sugar, okay. Now it is linking another phosphate and this phosphate is linked to the 5 position of the another you can say nucleoside, okay. So this is how they are linking. Again another third position of the sugar, the phosphate is present and this will be linked to another sugar at 5th position. So if you look from the bottom to top, you will find 3 and 5 dash, okay. So you'll find 3 dash, 5 dash, 3 dash. Now why this dash is used? Because we assign 1, 2, 3 number to the ring that are present in the nitrogenous bases. So in order to number the carbon in the sugar, we use 1 dash, 2 dash. So we say that it is 3 dash, 5 dash. But if you look, since the DNA is made up of two different strands, so if you look at the another strand, Okay, it runs in the anti-parallel direction, just opposite to it. Okay, where the orientation of sugar is facing toward the other side. So if you find the other position of bond is accordingly you can say placed 3 but the position 3 and 5 will remain the same. So once you look at the two strands of DNA, you will find one run in 3 5 direction, another one run in 5 3 direction. Now this is also important. If you see, if this orientation, change of orientation is not there in the other chain, then there is no bonding that exists between G and C and A and T. So in order to facilitate the bonding, hydrogen bonding and stabilize the structure of DNA, one strand is parallel and another one is anti-parallel. So there are 3 bonds that you can see between G and C, and there are 2 bonds between the adenine and thymine. Now James Watson and Crick, Francis Crick, 1950, they proposed the double helix model of DNA, and for which they were awarded Nobel Prize. So according to that, DNA consists of 2 polynucleotide chains, as we have seen, and then there is phosphodiester bond between the 2, you can say, sugars in one polynucleotide chain. So the 2 polynucleotide chain or the strand that spirally twisted around each other and coiled around to a common axis to form a right-handed double helix. 2 strands are anti-parallel to each other. One run in 5 3 direction and another one in 3 5 direction. At 5 and it has phosphate, at 3 and it has hydroxyl group. The polynucleotide has phosphodiester bond where the hydrogen bonds connect the 2 polynucleotide chain. This base pairing is called complementary base pairing, where one purine base pair with another pyrimidine. Okay. Now if you look at the dimensions, see, look at this structure. You can see the axis is marked here and width is marked, and one complete turn of helix is marked. Now here just I would brief you. There are different types of DNA, like as far as their orientation is concerned: A type, B type, C type, and their dimension varies. So this we are talking about common form, that is B type of DNA, which is present in almost all the human. And Watson and Crick, they work on this particular model. So I'll be discussing here. There are 10 base pair in one turn of helix. The width of DNA is 0.20 nanometer. The distance between 2 consecutive base pair is 34 nanometer. The length of one complete turn is 3.4 nanometer. The total length of DNA is 2.2 meter. The dimension of diameter of nucleus, as you know, is 10 raised to minus 4 meter, whereas you can see the length of DNA is 2.2 meter. So how such big you can say DNA fits into 10 raised to minus 4 you can say diameter sphere? Okay, so it means what? There has to be some organized packing takes place in DNA, then and only then such large structure will fit into the nucleus. So DNA packaging, let's talk about that. The term chromosome was introduced by Valdier in 1888, and the DNA is packaged into the chromosome with the help of the protein called histones. Histones are actually basic protein which are rich in basic amino acids such as arginine and lysine, and they are positively charged because they are basic protein, and they stabilizes the negative charge of DNA. Okay, and if you see, there are total 4 types of histones: H1, H2, H3 and H4. Now if you look at H2, H3 and H4, 2 molecules of H3, 2 molecules of H4, and again H2 is of 2 types, H2A and H2B. So 2 molecules of H2 and 2 molecules of H2. Together there are 8 molecules of these and they form the octamer core. Okay. So H1 is not part of the core. We will see how the structure is organized. So DNA is wrapped around octamer of histone to form nucleosome. Okay, so this whole combination, that is combination of DNA with octamer histone, is called nucleosome, and one nucleosome core. Now what is nucleosome core? No, nucleosome core, which exactly consists of DNA and the octamer. Okay, separated from other by linker DNA. Okay, so linker usually is of 30 to 60 base pair. Now just look at here. First I'll make you understand this one. Okay, now see, from here to here this is called as nucleosome core, but if you look at from here to here, this portion is called nucleosome. Okay, nucleosome has histone 1 plus this represent the histone octamer, which consists of H2, H3 and H4, 2 molecules of each. Okay, and this pink color is the DNA which wrapped around the octamer. Okay, so this portion where the octamer is binded to this much of DNA is called nucleosome core, whereas H1 is located outside the core, and this portion of nucleus, if this whole nucleus, this is core, so whatever is the remaining portion, this is called linker DNA. So now if you look, the DNA in nucleosome is 200 base pair, DNA in nucleosome core is 140 to 166 base pair, and that linker is of 30 to 60 base pair, and octamer formed of 8 histones. Okay, now this kind of arrangement usually you will see, this is called uncoiled stage of chromosome, usually seen in the interphase stage. You have read about the cell division. So during the interphase stage you will see this is also known as chromatin network or bead on string structure. Now next step is further coiling of nucleosome occurs. So further coiling occurs, and this coiling, now it is converted into chromatin fiber. It further condenses, again some organized form of coiling occurs, and we say that chromatin is now condensed. The highly condensed structure of this you will see in metaphase, where you will see duplicated structure of chromosome. You can see centromere and its arm, and the one arm, this arm is called sister, one chromatid arm, and these two are called sister chromatid, you can say, and this one part is called arm. Okay, so this is called duplicated structure. Now here you will see a spot called kinetochore. It is a protein structure that form on the centromere during cell division, and it links the chromosome to microtubule, that is the spindle. Okay, so this is something we have discussed in cell division. Now let's come to, so therefore the eukaryotic chromosome is structured. What we refer to the structure of chromosome, since you have seen chromatin fiber, duplicated chromosome, and then beads structure. So all these different names are actually given to the chromosome only. So it refers to the level of packing that occurs in the chromosome. So chromosome is represented like this, and this is the duplicated structure of chromosome. Duplicated means the replication of DNA, it occurs in the synthetic phase of cell cycle. So the information of chromosome doubles, and this is called as duplicated structure of chromosome. Okay. Now chromosome constrictions. Centromere is the primary constriction of chromosome, but in some chromosomes secondary constriction is also present. Secondary constriction is also known as a nucleolar organizing region that help in the formation of nucleolus. The secondary constrict is called satellite region, and this serve as marker They serve as a marker to identify the chromosome, and the chromosome with satellite is called set chromosome or satellite chromosome. Now let us look at this structure. So this is the primary constriction. You will see secondary constriction. So the portion after the secondary constriction is called secondary constrict or satellite. If any chromosome has such a structure, we say this is a set chromosome or satellite. Now since it is different from others, so you can easily identify this structure and you can easily study and locate the genes. So that's why it is also known as marker. It is used as a marker for the study of molecular biology and genetics. Then you can see telomeres at the end. Now actually, what are telomeres? They consist of highly repetitive sequence of DNA that do not code for any protein but function as cap to prevent the chromosomes, to keep the chromosomes separate from fusing together, because chromosome is actually nothing but it is DNA. Okay, so repetitive sequence are present throughoutly. If you see in DNA, we will study more about these repetitive sequences, and these sequences are the one where the genes are the functional genes are not present. Okay, but some other things are present in these regions. We will talk about that later, and they are usually show very less expression. Okay, now again, if we talk about the satellite DNA, see here I was talking about satellite chromosome, but there is a term called satellite DNA. I'll just to make you understand that satellite DNA is different from satellite chromosome. Again, we'll be reading some details of satellite DNA in some other chapter. Now it refers to tandemly highly repetitive sequence of the stretch of DNA in large number of times. For example, tandem repeats like AATGC. Okay, and just after that again it will repeat, so it will repeat subsequently AATGC, 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 AA. So this is called tandem repeat. So such kind of sequences are present in the satellite DNA, and mostly it is located in telomere and centromeres region of the chromosome. So they don't carry much functional genes, and this is also known as non-coding DNA. Again, we'll be talking about non-coding DNA in the next video. Now also look at the types of chromosome based on the position of centromere. So telocentric, acrocentric, submetacentric and metacentric. Telocentric centromere at the terminal position, acrocentric centromere near extreme end, one arm looks short and one very long. Submetacentric centromere is slightly away from the center, and metacentric centromere is in the middle and all the arms are equal. Now on the basis of staining, the chromosomes are divided into two regions. Hetero, like this is on the basis of staining. So if you have the chromosomes or DNA, then you can do the staining and you can study the chromosome. So when you stain it with acetocarmine or full gene, you will find that chromosome take up the stain in different way, like, and it is divided into two regions, heterochromatin and euchromatin. So heterochromatin is the darkly stained region, and this region is highly condensed and transcriptionally inactive. It consists of highly repetitive sequence and less sensitive to mutation and resistance to nuclease digestion. Whereas if you look at the less darkly stained, which is called euchromatin, it consists of loose DNA which has transcriptionally active region. So this is all about chromosome and DNA and chromosome. Thank you.